Nanopores are used in scientific instruments to identify molecules, like DNA. Here's how it works. You pass your molecule through a tiny hole, the nanopore. And as it passes through the hole, it generates electrical signals. And those electrical signals form a unique fingerprint that lets you identify the molecule.
Which sounds simple enough, right? Of course, even rocket science sounds simple if you want it to. Just put fuel in a big tube and set it on fire and point it at the sky. Just like rocket science, solid-state nanopore research sounds simple but is actually insanely hard. First, there's a vast ocean of motion. Brownian motion, rotational motion, intrinsically disordered motion. Even when molecules get sucked through the pore, they don't file through in nice civilized queues. They bounce and jostle like crusty punks in a mosh pit in a crazy molecular hurricane. It's like you're trying to take a photo of someone spinning 10,000 times a second, and you need them to sit still. And your first thought might be, well, why not just use a super fast camera that is faster electronics with a higher sampling rate? Sure, that's a knob you can turn, but it brings another humongous problem. Electronic noise. So many types of noise. Flicker noise, protonation noise, Johnson noise, shot noise, dielectric noise, capacitive noise, amplifier noise. Even when you model the physics perfectly, the math delivers a brutal verdict. You cannot just outrun the noise. Turning up the speed knob also turns up the volume on the interference. We need to be able somehow to control how molecules move when they mosh pit hurricane their way through our pores. Most nanopore research at the frontier is all about this problem: how to achieve translocation control. One method is literally called ping pong. You pass the molecule back and forth repeatedly so you can combine multiple measurements. This sometimes works heroically well. Henry White's lab at the University of Utah achieved calibrated precision equivalent to less than the volume occupied by a single hydrogen atom in Edwards et al. 2015. More recently, the Nivala group at University of Washington played ping pong with single protein strands in a biological nanopore. They demonstrated proof of concept for single molecule reading and analysis of long intact protein strands with single amino acid sensitivity. This was heroic. But the dirty little secret of frontier nanopore research is that we talk about papers like this as heroic efforts, because they are indeed heroic. You're looking at thousands of hours of tedious, painstaking, hands-on experimentation and planning for one very cool paper like this. We know all about that struggle at Molecular Reality Corporation because early on, we developed a new form of translocation control in the form of mechanical nanopores, apertures formed by mechanically sliding one piece of silicon back and forth over another. With this, we could capture and oversample individual nanoparticles, and early results suggest this will eventually provide a viable path to the holy grail of solid-state nanopore-based DNA sequencing. But if you tried to fully develop and commercialize it in a siloed, traditional biotech startup setting, you could easily spend $500 million and still fail. That's a truly systemic issue that plagues all of biotech toolkit research. Limited funding and attention means we all have to flex our egos and fight each other over intellectual property. We must do things differently. And here is how. We are building solid-state nanopore R&D devices that are as easy to use as toys, so people can quickly and easily try out different things. And we are coordinating everyone with instrument control software that turns data collection into a game. This is how we will find the ideal nanopore methods that will allow us to achieve universal molecular streaming, a world where anyone can easily see anything that exists in molecular reality. Your membership will put you on the front lines of this generation-defining mission. Join us in our epic quest, and remember, biology is just beginning.